So I'd like to start off by thanking Nevan and Trey for giving me this opportunity. I really enjoyed all of the talks. So our lab is interested in studying host pathogen interactions from the point of view of bacteria. There are several intracellular bacteria that infect humans, and as you can see from some of the examples that I've shown you here, these bacteria can access various subcellular localizations inside the cell. Some go to the cytosol, some will engage the Golgi, some go to the ER, some to the endosome, and some can even survive the acidic environment of the lysosome. So when you look at these bacteria, the first thing that comes to mind is that they cause a human disease. And a lot of these bacteria develop antibiotic resistance, so it's important to know how they cause the disease to get to better therapeutics. But what I am interested in, because of my cell bio background, is I see these bacteria as amazing cell biologists. And the reason I say that is, naively speaking, we are apparently the most evolved species on the planet with 20,000 odd genes, as you've heard many times today. And yet these bacteria, they have less than maybe 100 proteins that they're secreting in our cells, and that's enough to kill us. Why is it that we even need an antibiotic or a vaccine to fight them? Why can't we just fight these bacteria? They're so much smaller than us, so many less genes. So this is a sort of a fundamental naive question I had, and I just dug into this field coming from a cell bio perspective. And one thing I think is one of the answers to that is that most of these bacteria, they divide every 20 minutes. And we humans, maybe once every 30 years. So we have a disadvantage in that way because we can't mutate our genes and evolve as fast as some of these bacteria can. So out of all of these pathogens, I chose to work with this one called Legionella. Legionella was discovered in 1976. There was a conference in Philadelphia on the American Legion, and a lot of these people were ex-war veterans, and they were staying in a particular hotel in Philadelphia. And because they were more elderly people, a lot of them got a severe type of pneumonia, and many of them died. And at that time, people thought this was the first act of bioterrorism. Turns out it was not. It was that the bacteria had contaminated the cooling system of that hotel, and as a result, people were inhaling contaminated aerosols. And that's how Legionella first struck as a human accidental pathogen. Now, out of all the pathogens that I showed you in the previous slide, most likely Legionella infects and kills the least number of people. It's not like I would like to kill people, but it's a little bit of a challenge to explain why is it that you want to work with a bacteria that doesn't kill as many people. And the reason is that Legionella has already turned out to be a great tool for discovering cool cell biology inside the cell. So Legionella has many unique roles in biology. So one of the things that we found from studying a Legionella effector was that it was causing a new type of post-translational modification known as phosphocholination. And it turns out that the domain that this bacterial enzyme was using to cause this new modification is conserved not just in bacteria, but has one copy in every single metazoan species, including us humans. And our protein is called Huntington interacting protein E. And this protein has this fixed domain that this bacteria is now using to cause this new modification. So by learning something about an enzyme from a bacteria, it's not surprising that we can learn really interesting cell biology into higher mammals. The next example is that this bacteria can encode an effector or a protein that actually is a histone methyltransferase. You heard from Jan today about the SED domain. So this bacterial protein has a SED domain. And so just imagine a bacteria has no nuclei, has no histones, and yet it has its own histone methyltransferase. So it doesn't even need the host enzyme. It can use its own enzyme to alter the transcription of the host genome. And not just that, the lab next door to us worked on histone methyltransferases, and we gave them the enzyme to test its activity against the mammalian enzyme. Turns out that the enzymatic activity of this enzyme is 1,000-fold better than the mammalian homologue, and that explains why it can win these competitions between host and pathogens. The next example is a very unique case of ubiquitination. So we know that the ubiquitin dogma in the field is that ubiquitin is a modification which is transferred from an E1 enzyme to an E2 enzyme, and then finally to an E3, which is a ligase that adds ubiquitin to the substrate. And these processes require ATP. Legionella has an enzyme that has an ADP ribose transferase domain, and it doesn't even need E1 and E2 and can directly modify ubiquitin itself. So it can modify a modifier and then use that to directly add ubiquitin to its substrate. Now it turns out that mammalian cells also have this domain. And so now the search is on as to whether we have missed all of these ubiquitination events because we didn't look for enzymes that have this particular domain. And so these are only the 3 examples I talked about where post-translation modifications are involved. But in the broader picture, studying Legionella and the cell biology of Legionella has really taught us a lot about how bacteria and the host are engaged in an arms race. So the question is, what else can we find by studying this bacterial pathogen? And to understand that, we have to first look into the life cycle of Legionella. So Legionella enters the cell in a plasma membrane-derived vacuole. It uses its type 4 secretion system to secrete close to 300 bacterial proteins inside the host cell, a lot more than the regular intracellular pathogen. And although we don't know the function of most of these effectors, we can broadly categorize them into 2 categories: the negative regulators, which block the trafficking of this vacuole to the lysosome to prevent its degradation, and the positive regulators, which slowly hijack these ER-derived vesicles and coat this plasma membrane-derived vacuole and make it resemble like an ER. It even has ribosomes around it at 8 hours. And once it's reached that point, it will replicate and then will lyse the cell and come out. So the key for Legionella is to do 2 things. First, it has to protect itself. Intracellular bacteria has chosen a really dangerous life cycle. It is going and staying in a host which is out there to kill it. So what it has to do is to protect itself from the host, but at the same time, it cannot kill the host because it needs the host to replicate. So as a result, these intracellular bacteria often engage in a warfare that's tightly regulated, and for that reason, the effectors that they use are really interesting tools to study host cell biology. So this is the cartoon, and this is the real image, and you can see here in red, each single red dot is a bacterium, and you can appreciate that when the Legionella-containing vacuole forms, there's actually a lot of bacteria in the cells. So for that reason, it means that it knows how to expand this ER. It knows how to gain more membrane because it's gaining in size, and it's really manipulating a lot of these processes. So we are interested in studying most of these processes and as to how Legionella can steal ER vesicles that are en route to the Golgi and instead make them fuse to a plasma membrane-derived vacuole. Remember, the plasma membrane has snares which are not compatible with these ER vesicle snares. So the fact that these ER vesicles are going and fusing there is already poses a problem of canonical snare pairing. So all of these processes are worth understanding to know how basic cell biology mechanisms work. Now, when I say we want to use Legionella as a tool, what do I mean? So I'll give you an example today of just post-translational modifications And you know that there are only like about 518 kinases in humans, and yet when we look at the phosphorylation databases, there are more than 275,000. So there is some kind of disconnect in looking at these numbers. Either some of these phosphorylation events may be promiscuous and they don't really mean anything to biology, or they might mean something and we're not really figuring out how to look at these modifications in a proper way. So we asked the question, can we use a pathogen to test this idea that if a phosphorylation is really important and is being utilized by the pathogen, it might give us a new role for that particular phosphorylation or these other modifications. And with the collaboration with Nevan Krogan, we looked at all of these modifications during Legionella infection, and we took a sort of a broad approach. But at the same time, we also had this idea that maybe we can do in parallel a more directed approach to look at these modifications inside the cell. And so for that, we collaborated with Keyvan Shokat's lab at UCSF, who is an expert in protein kinases. And we had a joint student, Stephen Moss, who's a chemistry and chemical biology student. And what we asked is, what does these Legionella kinases that are encoded by Legionella do inside the human cells? Now, mostly bacteria encode histidine kinases. They don't encode the serine threonine kinases, which are more like eukaryotic kinases. But Legionella in its genome has 4 serine threonine kinases that look like eukaryotic kinases. And one of them has an uncharacterized domain attached to the kinase domain. So the question I had was, what are these proteins doing in Legionella when they look like eukaryotic proteins? And the answer, I thought, would be that they would be hijacking some machinery that's important to the cell. It must have stolen these kinases from some of its eukaryotic host and now using it against us. And so for that reason, we looked at this particular kinase called Legionella kinase 4. The first thing we did was we expressed it in cells, and we saw that all the cells that were expressing this Legionella kinase in a GFP expression vector showed a Golgi fragmentation phenotype. All of those cells showed Golgi fragmentation compared to untransfected cells. When we made a kinase dead mutant of this protein, we saw that now the Golgi was normal, suggesting that whatever the kinase activity was disrupting host trafficking such that the Golgi was getting fragmented. Next, we ask, is this modification deleterious to the cell? How does the cell react to that? And for that, we took advantage of yeast. We put it in a galactose-inducible promoter, and we expressed this LEC-4 plasmid, and we saw that the yeast cannot grow in the presence of this bacterial protein, suggesting that this bacterial protein, whatever it's doing, is not good for a eukaryotic host. And even though yeast is not a natural, Legionella is not a natural pathogen of yeast, it can do something to yeast that is not allowing these yeast cells to grow. So the question is, how can we find the target of this kinase and answer the question, why is it so lethal to yeast? Now, what we did was we utilized a system that Nevan's lab has pioneered. So what they do is basically they take the kinase domain of the enzyme in question, and they make a mutation so that now it can increase the size of the acceptance of ATP. So as opposed to just ATP gamma S, now this kinase is able to accept these larger analogs of ATP. Now just imagine if you put these analogs inside the cell, all the endogenous kinases, those 519 kinases, will not be able to accept these because they have smaller kinase domains. But the only one that will accept it is LEC-K4 because we made a mutation and increased the size. So now all of these analogs will be incorporated. And so now we can do an assay and show this nice labeling, which you don't see with ATP gamma S, but only with LEC-K4 you see that. And so the question is, can we use mass spectrometry to identify what is the substrate of LEC-K4? We also saw that LEC-K4 was autophosphorylating itself, which is common to see in other kinases. So when we did mass spec, we were very sad because we saw that it was HSP70 family. And HSP70 is one of the most abundant proteins inside the cell. It comes down with almost all mass specs. And we were really sad to see that it was a chaperone and not some really cool proteins that are involved in Golgi fragmentation. But someone rescued us, and that was Nevin Krogan. There was a paper published from his lab, and I don't know if he still remembers this paper. I will ask him later. It showed that when they did post-translational modifications in 12 different organisms, ranging from yeast till humans, they found that a lot of the post-translations modifications that we find in the cell there is a lot of noise. But there are some that are specific, and they're conserved between yeast to mammals. And one of the proteins that they discovered and studied more in detail was HSP70. So when we saw that, we were like, it's possible that HSP70 is phosphorylated in yeast, and that's why we are seeing that yeast lethality phenotype. So let's go deep and see if this is actually a real modification. And when we purified it, we got robust phosphorylation with two members of the family, HSP70 and BIP. And then what we did was we looked at other HSPs, HSP90, and we saw that HSP90 was not being targeted, even though HSP90 is another really abundant protein. And the other thing we saw was that the E. coli HSP70 was not targeted, but the mammalian ones were. When we changed the residue, we saw that actually eukaryotic HSP70s has a threonine in it. The corresponding bacterial one has a serine, which explains that why Legionella, being a bacteria itself, is not able to cause its own toxicity. And we could change this threonine to serine and show that even though it's not targeting its own molecule, if you mutate the host molecule, it can still, to some extent, cause phosphorylation. And we noticed that HSP70 phosphorylation changes its ATPase activity. So a chaperone uses ATPase activity to fold proteins correctly. We looked at the protein folding capacity, and we gave it unfolded luciferase and allowed it to fold. And we saw that all these phosphorylation was blocking the ability for chaperones to fold. The question is, why target a protein that is just innocuous? It's folding proteins correctly. What would the bacteria gain from that? And we noticed that during infection, actually the phosphorylation happened on HSP70, but not BIP. Remember, BIP is in the lumen of the ER, HSP70 is in the cytosol. So I think that the effector is coming to the cytosol and is targeting specifically proteins that are of the HSP70 family in the cytosol. Next, we also looked to see what is the consequence of HSP70 phosphorylation. And in our previous paper, we knew that the unfolded protein response, which is caused by adding tapsigargin, is completely inhibited if you have wild-type infection, but not with a strain that lacks the secretion system. So you don't see an increase in BIP and CHOP when wild-type is infected. And so for that, we thought maybe it has something to do with protein translation, and maybe this phosphorylation is affecting protein translation. And indeed, what we saw was expressing LEC-K4 in cells completely blocked the expression of BIP. But even when we added tapsigargin, we didn't see the expression of this chaperone. So, we were not sure whether this block in protein translation was only to BIP or to the global protein translation. So when we looked at global protein translation with the help of this assay, where we add propargyl glycine to the newly translated polypeptides and do a click-it reaction, we noticed that LEC-K4 cells were not causing any protein translation, whereas the kinase dead were. So this gave us the idea that somehow this chaperone phosphorylation leads to blockage of protein translation. So what is the link between phosphorylating a protein, chaperone, and then blocking protein translation. We looked at the literature and we found that these heat shock proteins actually, they are associated with the nascent polypeptide change on the ribosome. And when you heat the cells, what happens is they fall off from the ribosomes, and that blocks protein translation. So we asked the question, is Legionella blocking protein translation by phosphorylating this residue, which then allows HSP70 to fall off the ribosome? And indeed, what we noticed is that compared to uninfected cells, wild-type infected cells had less HSP70 associated with the ribosome fraction. And to drive home the final point, when we looked at the polysome profiles, we saw that all the HSP70 that's phosphorylated was mostly in the fractions that was not associated with the ribosome. So now the take-home message is that we have uncovered a single modification in one site of a protein that can globally block protein translation. Now, this is not uncommon for bacteria. The first toxin that was identified from bacteria is tetanus, the ADP ribose toxin that diphtheria uses to block protein translation. But whenever we talk about protein translation inhibition and toxins that cause that, we oftentimes have only uncovered the protein translation machinery itself. This is the first time where an innocuous chaperone, one single phosphorylation, is showing to block global protein translation. And because they found this in yeast and in mammals without causing any infection, we think there might be a kinase that can cause this. And so I would like to end by thanking all the lab, especially Stephen and Kevin, who were instrumental with this project, and all the collaborators and funding. And thank you for your attention.
How about phosphatases? Is there any seemingly eukaryotic phosphatases in Legionella?
Yeah, so we oftentimes, whenever Legionella has an enzyme that targets a host protein, the gene next door to it has the anti-activity, and it will reverse it because it tightly regulates it. And we did look for genes that are upstream or downstream of LEK4, and none of them are phosphatases. But we do think that it's possible that there is a phosphatase that we were not able to identify yet.
No question. So you mentioned the yeast result. Did you look for HSP mutants in yeast? Can they overcome it?
Yeah. So in their paper, they showed that if you make mutation in that residue that is phosphorylated, yeast is lethal. So you can't make mutation in that domain. Okay. Thank you.